Good morning, everybody, and welcome to the first of our Living Soil series. Today, we're going to be joined by Dr. Sheila Storey and Dr. Ebrahiema Arendse. The Cape Town Together Food Growers Initiative is a collective of anybody who wants to grow food with a vision to create a food forest out of Cape Town to enable food freedom. It originated during the pandemic to connect the entire Cape Town and surrounds. The Food Growers Initiative is hosting this event, which is curated in collaboration with the PHA Horticultural Area Campaign. The Living Soil series is kicking off today with a deep dive into the basics of soil biology. This will cover what is soil health, making the biology work for you, how to improve the biology, the 5 soil principles, and how to measure the biology. We hope this is only the start of a conversation that leads to action, to regenerate the soil with organic waste and expand the urban options and applications to unlock further innovation and entrepreneurship through collective action. Today, I'm going to ask Nazeer Sonday of the PHA Horticultural Area just to say a couple of words. Nazeer, over to you. Nazeer, you're on mute. But we can't hear you. Do you need to put your headphones back on?
Okay, what about now? Can you hear me now?
Yes, we can. Thank you.
Okay. Can you hear me?
Yes.
Great. So we've got a bit of a sound issue here, but we'll figure it out as we go along. Great. So I want to say good morning to everybody. I'm happy to be here as the campaign, the PHA Food and Farming Campaign, but also as members of the Verkop Polyculture Farm. Behind me, you'll see some of our farmers on the farm. Can you raise your hands there? Great. Feedback. Okay. Let me continue. So I'm happy to be here and collaborate with the Cape Town Together group. And I'm hoping we're going to have an amazing soil workshop series. I want to just quickly say that, we have been, as a campaign, we have done 2 soil workshops, one in 2017, one in 2018, where we tried to share and learn about the soil. And in these workshops, we had our own soil scientist, Sheila, Storey, prominently featured in these workshops. So I'm happy to, in this year, during the pandemic, that we can again do this. We know of lots of soil scientists like Elaine Ingham and Christine Jones and Ray Achuleta, but we have our own homegrown soil scientist, Sheila Storey, who runs a lab it's called Nemlabs, and we're very, very happy and very, very lucky to have amongst us. I want to quickly say that this is an example of how science and indigenous knowledge comes together, and in this case protect our soils and create more understanding of our soils, and then as farmers learn how to regenerate our soils, which has been historically been degraded. We in the PHA understand soil very, very well. We understand that we need to change the way we farm because we have an amazing aquifer in our soil, and we need to farm in an aquifer friendly way. We need to increase our biodiversity. We have huge losses in our insect and plant species, which we which we want to see and regenerate. And of course, we see the soil as a medium to do that. And for us, soil is very, very important. With that few words, I want to hand over to Erika. Thank you, Erika, and I hope everybody enjoys the workshop.
Thank you very much, Nazeer, for those words. Without further ado, let me introduce Sheila Storey. Sheila holds a BSc in agriculture, a majoring in entomology and plant pathology at Stellenbosch University. This was followed by a stint as a researcher for the Department of Agriculture, working on maize in Potchefstroom. She transferred to Stellenbosch and worked on small grains, example wheat. After completing her MSc in nematology, she lectured at UWC in a course geared at crop protection. In 1987, she started the Nematode diagnostic laboratory Nemlab, which now resides in Klapmuts. This has expanded to include labs in Uppington and KZN. She consults all over Southern Africa, covering all crops. In 2016, she was part of an initiation of the HortiGrow Soil Health Workshop and involved in various research projects, looking at using nematodes as bioindicators of soil health. In 2020, she took over the Soil Health Support Center, which now forms an integral part of their laboratory. They also do insect IDs and have started a plant disease laboratory. She travels all over Southern Africa doing presentations on nematodes and soil health. Welcome, Doctor, and the floor is yours.
Thank you, Erika, for that introduction. I must also thank Nazeer. It's a pity I can't be there. I enjoyed my 2 previous visits and interacting with the farmers. I just want to make 2 corrections. The one, I don't have a doctorate, although most people think I do. I only had a master's, and then I opened my own business, and that kept me busy for long enough. The second one, Nazeer, I'm not really a soil scientist, although I'm having to learn more and more. I'm what they call a soil ecologist, but even there, it's such a wide field. I prefer to just stick to the nematology, but we are expanding. Okay, I'm going to share my screen. And with that, we hope to start. Okay. Can you see, Erika? Are we going?
Yes, you can see your screen.
All right, so as an introduction, and what Nazeer and Erika have asked me to talk about is living soil and the basics of soil biology. In a teaspoon of soil, you can find so many organisms that they're in fact more than what you have people in the world. So please understand, I'm really just going to touch. It's a very complex subject, but what I'm hopefully trying to do is just show you what is soil biology, because you keep hearing words and how it works. I think that's the most important thing, is in a way to demystify or demyth it. Yeah, let's see how far I can get with this. Oops, sorry, don't know what I did there. So I'm going to talk about a simplified soil food web. For those of you who follow Elaine Ingham, we have our own one which we've developed here. I'll talk a little bit about life in the soil and give you various examples, but very few. As I've said, it's a very complex system. I'm going to talk about nutrient cycling, try and unpack it to why the organisms are important and what that we mean when we say nutrient cycling. I'm going to talk about soil health, but I think a lot of you know more than enough. Those of you who've been working for a while on your plots or your farms. And then how do we achieve soil health? The 5 soil health principles that Erika mentioned. And then hopefully at the end, I'm going to talk about measuring soil health, which is what we do in the laboratory. Okay, to start off with, if we look on the left-hand side, the soil consists mainly of mineral particles and a thin layer of organic material. So your purpose is to increase that organic material or the humus and make that little, if it's a purple layer, to increase that, make it deeper. In that organic material, there are various things, and we can start at the bottom: the stable organic material or the humus, the composted organic material, which is still in the process, fresh residue, which you've added to the top, And then in that is the living organisms. And I've just sort of mentioned a few. Earthworms are the ones you can see. Sometimes you can see the fungi as the hyphae, those white strands. Bacteria you definitely will not see unless you see like a pus which is developed. Arthropods are the little insects in all shapes and all sizes. The nematodes you definitely won't see. You can sometimes on your plants, on the roots, see galls. That's a very specific nematode. And I can perhaps mention, yeah, I've spoken to Erica that after this basic biology, we will move on to the nematodes and fungi and bacteria, which are harmful, which are affecting crop production. But that's for another day. And then the last one is the protozoa, and you definitely cannot see them with the naked eye. So the problem that we have with the soil biology, it's microscopically small. To show you pictures on the microscope, so at the top is the fungi and bacteria. The fungi on the top left hand, the bacteria on the top right hand. You can say we've just odd ones which we've just taken photographs of or got from the web. And just to give you some examples, so it's a huge variation. The bottom left-hand side is the protozoa, the amoeba, the ciliates, and they're lovely to watch under the microscope. I can show you a video now. They move around at great speed. And then the predatory mite, and then the bottom one, the springtail, and that you can sometimes see in your garden when you've got a lot of organic material. They're like little white things that bob up and down, but they're minute. And then on the right-hand side in the bottom corner is the nematodes, and you get those that are harmful and those that aren't. And yeah, we will be discussing them because that's my speciality, and that's kind of what I'm going to concentrate on, if you don't mind. I'm going to try and do a video, just what we saw under the microscope on Friday. Can I, if you don't mind, I'm just going to open a video and I hope it works. There you are. Sorry. It's not showing. Yeah, it's not showing. Sorry. I don't know how to. Oh dear.
You can stop sharing your presentation and then go back in and just share the video.
Yeah, I think that's what I'm going to do. Sorry, I'm just. Please be patient as I try and try again. Can you see it now? Oh wait, I didn't share it. Sorry, I will get there eventually. Connect to Mockdown.
Well, Sheila, is there a meeting?
Share screen. I'm going to try again. Share screen, that one. No, okay, sorry, Erica, I was hoping to do it, but it's not going to work. I'm going to, because it didn't stop sharing, that's why. Back to where we were. No, I'm not. Sorry.
What you can do is you can send me the video via wire transfer, and I'll send it out after the meeting.
Okay, we can try and do that. All right, let's move on. So I've tried to show you, but you know, not what's on the microscope. No, it won't. Okay, so back to where we were, the soil biology. This is just a schematic drawing of what I showed you the microscope slides of. So bacteria, earthworms, protozoa, but look and see at the earthworms, we call them shredders and grazers. Some of the arthropods are also shredders. They're responsible for the breakdown of your organic material. So you definitely need them. The fungi are divided into mycorrhizal fungi, saprophytic, and also predaceous fungi. So yeah, it's a huge group. The nematodes is what I'm going to concentrate on because that's the group that I know. And then the predator arthropods, those that feed on other predators. So just to show you what happens underneath the soil level, the brown line in the middle. So above ground, you know what your ecosystem service providers are, your plant pollinators. You've got your shredders and decomposers and your biological control. But under the soil, there are also a lot of ecosystem service providers. Your earthworms are responsible for soil aeration, your smaller ones for nutrient cyclers, and that's what I'm going to try and show you, is what they do, how advantageous they are to you as a farmer and why we need to keep them. This is our soil food web. It's a little bit different to Elaine Ingham's one, but what I want to show you here, the most important thing out of this whole diagram is the top layer. They're different trophic or feeding levels. So the first or the second trophic level at least is the decomposers, the mutualists, and the pathogens, parasites, root feeders, that group. The second one is the shredders, the predators, and the grazers. You can see they're a little bit sort of one step up in the food chain, and then you get your high-level predators, and then again, eventually you get to even higher, those that are above ground, like your mice or your birds or whatever. What's interesting and what I want to point out is that nematodes are on the second, the third, and the fourth trophic level. You find them everywhere, and that's why they're such useful bioindicators of soil health. I just want to put this in because it's very important to me. South Africa has a very high level of soil biota diversity, which is unique to us. And for that reason, and I'm passionate about this, please don't bring in external biology from what's imported. Let's use our own stuff and make it work for us in South Africa. So the biology examples that I'm going to show you are the Collembola or the springtails. They're part of the arthropod group. I'm going to touch a little bit on the Mycorrhizae. It's a very special fungi. You can see at the bottom, there's some pictures from my laboratory, and then on the nematodes. I'm not even going to touch on fungi, bacteria, protozoa, mites, or earthworms. It's just simply too much to cover in an hour. So your springtails are small, they're abundant, they're quite important for soil structure, aeration, and decomposition. And they're also very important prey for predators. So they sustain the natural numbers of the pests and they keep them low. We have a lady at UCT. She joined them last year, and she's a specialist on Collembola in South Africa. So the direct ecosystem service providing provision that they do is the decomposition of the organic matter and the recycling of nutrients. So they consume the dead plant organs which are on top of the soil. Also very important, they consume the excreta from other invertebrates, and I'll talk about that later again. They also regulate soil microorganisms, but very important, they spread the fungal spores and the bacteria through the soil. And their fecal pellets provide a favorable habitat for other organisms. And that makes them important, an important, what would I call it, link in the soil food web. So if we look at the Mycorrhizae, it's a fungi that colonizes plant roots, and they're quite common in the soil. We found them in research in the apple orchards and in the vineyards. They live in symbiosis with 90% of our plant families, and the exception is the brassicas. So if you're going to have brassicas in the winter, you're not going to have Mycorrhizae. They don't colonize the plant roots, but they don't disappear. They stay in the soil until you plant a host again. They form structures that improve the nutrient uptake, and we refer to them as arbuscules, which arbuscule is short for arbor, or it's derived from the word arbor for trees. And you can see in the middle, the picture there, the little trees that are formed inside the cells, and that's used to increase the uptake. The vesicles on the left-hand side are these round bags, and those have nutrients inside, and they store them inside the plant. And then the hyphae are these little brown lines in this, what would I call it, in a diagram, and they move between the cells and inside the cells and then outside the roots. Just also a diagram you very often see on the internet. On the left-hand side is a non-mycorrhizal root. It's just the root. It's got no mycorrhizal fungi colonizing it. And on the right-hand side, you can see it. And it's immediately you can see you've increased your area of absorption for nutrients, for water, and whatever else. Okay, so the very, very important aspect of the avascular mycorrhizae and why they're so important is that they provide the plants with phosphorus. They're actually responsible for the mining of the phosphorus. In return, the plant will give it carbon. So, and then you've got this carbon buildup in your mycorrhizae. So there's the first nutrient cycling that takes plants in the soil if you have the mycorrhizae present. So the key advantages, I've alluded to some of them. It enlarges the effective root absorption area. The absorption increase can be up to 50%, and you saw that in the photograph. It improves drought tolerance, and I'm going to refer to the picture at the bottom. It was an experiment done, so you can see the roots are the thicker parts in the top of the root compartment. And then as you can see lower down are the hyphae of the mycorrhizae fungi. So it goes quite far down and it can mine, if I can use that term, the water and supply it to the plant. So very important. They obviously increase your yield because of the huge advantage to your plant. It provides the plants with the phosphates in exchange for carbon in the form of sugars, which I also described. So you've got a carbon sequestration immediately. It produces glomalin, which is a glycoprotein, which is produced by the hyphae and the spores, which makes it forms these stable compounds that bind and protect your soil aggregate surface. And so if you don't have the mycorrhizae, you're going to have compaction. It's very, very important. They're also resistant to destruction, but mycorrhizae are very, subjected to plowing, destruction if you plow. You break them up very, very quickly. You lose those hyphae strands. So no plowing, please. We'll get to that. It's one of the 5 soil health principles. That was the mycorrhizae fungi. We turn to the nematodes. They're a diverse group that occupy the groundwater. They're unsegmented, as you can see in the pictures below. The size varies from microscopic to a few millimeters. In fact, they're all microscopic unless you can remove them from the soil. They're bilaterally symmetrical and the shape varies, mostly thread-like. And then sometimes, in the case of some of the females, they will swell up. And in the middle is a citrus nematode. The one that you will know as gardeners is the root knot nematode, which forms these huge galls on your roots. And then what's probably the most important point about the nematodes is they feed on various organisms in the soil. And I've shown you in this diagram with some of the pictures on the right-hand side, which we've taken in the laboratory, not colorful, we've made them colorful to distinguish them. Is the very basic trophic level is the nematodes that feed on yeast and algae and lichen. The bacteriivores are probably the most prominent. They have a hollow tube, you can see at the top there, and like these wings around their mouths, which they then almost circulate and then suck in the bacteria, so they feed on bacteria. The fungi have a thin stylet, you can just see it, it's very thin. It then penetrates the hyphae or punctures it and feeds on it. The next one is the herbivores or the plant parasites. It's got a huge stylet with knobs on it, and that actually punctures the plant cell and then either injects a toxin or extracts plant cell, and is very damaging, which you no doubt have come across. And then the omnivores are those responsible for feeding on organic material. They're also quite big. Then the second last group is the predators, and the predators eat other nematodes. There are lots of YouTube videos where you can see they take them and they suck them in. It's very dramatic, but they're quite scarce. You don't find them very often, but we don't want them in large numbers either. So mostly in a very healthy soil, you will have predominantly bacteriivores and fungivores, and then a few of the others. The last group is the EPNs, or entomopathogenic nematodes. I am trying to multiply them. We've got to doing the science, and we've got 15 million rand from the government. but now the legal system has put a stop to that. We can't get through the hoops, but quite exciting. So these nematodes are responsible for killing insect stages in the soil. That's for another day. All right, just to look at a closer look, you can see the bacteria forward in the top left-hand corner. They feed on the bacteria using those extensions and making wave-like movements. And then there's another picture on the top right-hand of the herbivores or plant parasites with that very strong stylet. And with a pump action, it will penetrate the plant cell. This one in the bottom left feeds on fungi. As I said, it pierces the hyphae. And then on the bottom right is those that feed on other nematodes with this tooth-like structure, which it has to sort of feed through the nematode that it's feeding on. Okay, so nematode functions in the soil. They're responsible for the breakdown of organic material. They redistribute minerals and nutrients, and we'll see that when I get to the nutrient cycling. They're in fact responsible for up to 30% of mineralizable nitrogen in the soil, and that's quite a lot. In other words, you can cut your nitrogen by almost 30% if you can get this to the perfect point. They sequester carbon through the feces of the fungi and bacteria that they survive on. They improve soil structure. They can regulate other diseases. As you can see, they feed on fungi and bacteria that could cause diseases on the plants. But as we all know, they're also responsible for huge economic crop losses. We've got to keep those numbers in check. In other words, you've got to keep this whole lot in balance. And then, as I've said before, the excellent bioindicators of soil health. And I just want to point out again, 30%, that's quite a lot of your nitrogen that you can provide. So how does it work? So the nutrient cycling, we can start off with a plant or a tree or whatever you have and the sun. And then what we tend to forget that the air that we breathe, 78% of that is nitrogen. And that nitrogen is free. We've just got to get it into our system. So the nitrogen from the air, the carbon dioxide also from the air, which we're trying to get into the soil, and the sun energy, we then have photosynthesis in the plant. And then the first set is the simple sugars, the amino acids, the organic acids and whatever. And those, the plant's roots will then pour out into the soil at the root exudates, and the bacteria feed on those simple sugars. This is a very simplistic diagram, but I'm just trying to help you through nutrient cycling. The next is the complex carbons, cellulose, lignin, tannins, which are root exudates. If the photosynthesis is working correctly, and on that group, the fungi will feed. In turn, they will then release back to the plant roots soluble nutrients, which the plant will then take up in turn. And that's almost your first cycle, if I can put it like that. The next group feed on the fungi and bacteria is the protozoa, and their excretions include water-soluble and root diffusate nutrients, again, which can be taken up by your plants, and that's your third cycle. The nematodes are the next group. They in turn feed on the protozoa, the fungi and bacteria, which I showed you earlier on. And that then, all of those, the feces or the actual bacteria, fungi, which have the nitrogen in them, and that is then released back to the plants. And this is where the nitrogen comes up, that is fed to the plants, and that you in fact need less in your soil if you can get your nutrient cycling going by getting the biology to work for you. I've put it in a simpler form. Remember this picture that I showed you in the beginning? So in the top is the fungi and the bacteria, and I refer to them as the fertilizer bags. The nitrogen is in there. Along come the protozoa, your amoeba, and they're the zippers. Remember, they feed on the fungi and bacteria. So they break open those fertilizer bags and release it. And then along come the nematodes, and remember, they feed on all of those, and they then spread the nitrogen through the soil and back to the plant roots as well. So it's a very simplified form of how to explain nutrient cycling. So we turn to soil health. I must point out that soil health isn't only soil biology. Please, it's your soil chemistry and your soil physics. And then the biology is just the third component of it all. So this is giving you all the advantages of what soil health will give you. But remember, soil is a non-renewable resource. If you can increase the organic material and you can increase the humus, it will act as a sponge for the water. So you have increased water holding capacity. But the other thing that the humus does, it forms a home for the microbe diversity. It needs somewhere to live in, and you've got to give it that humus. And through that, you've also improved your aggregate stability through the hyphae and the glomalin from the mycorrhizal fungi. So that's quite important. I'm not going to go to the details of soil health. You can read that afterwards. You get no crust forming. You get improved water infiltration. You've seen that. Reduced runoff and evaporation. I spoke to a milk farmer in outside Durbanville the other day, and he says he's got a dam which always filled up. He needs the water for the cattle, but he says since he's been practicing soil health, he's got no runoff and his dam won't fill up. So there's a very practical example of the fact that you can reduce your runoff. Improved nutrient cycling, and I've just gone through that and explained it to you. And carbon sequestration, you also saw that in the nutrient cycling that we worked through. The low weed and pathogen pressure. The weed pressure is a problem. The minute you've got open soil and that seed can germinate, it will. So cover your soil as much as possible. The reduced pathogen pressure is because you've changed the balance and you've got fungi and bacteria that are not harmful to plants, and you've got and feed on pathogens, and the same with the nematodes or the protozoa, but you've got to get that balance correct. So you've got a resilient system, and it is able to recover much easier the minute everything is in balance. So healthy soil is definitely the foundation of everything in a garden. How do we achieve it? And these are the 5 soil health principles we talk about. The first one is to cover it. And for those of you who know Nazeer, he has a hat like this. So that's why I use this as an example. Cover the soil in whatever format you can, be it a mulch, be it wood chips, keep it covered. The Americans have a way of saying, don't farm bare. Don't burn the microbes in the soil. Keep them covered. You're trying to keep the temperature down. The second one is diversity. And we have a saying, above ground diversity begets below ground diversity. And here I'm talking about crops, and I will come back to this on crop diversity and how we can use it. The third one is living roots. In that nutrient cycling, I showed you that you have to have roots because you've got to have photosynthesis and you've got to have the nutrient cycling. And so you've got to have living roots in your soil 24/7. Again, we get back no bare soil, please. Keep roots in the soil all the time. That's very important, otherwise you're not going to get the nutrient cycling going. The fourth one is reduced tillage. And we've actually, in the big trials we do in the orchards and the vineyards, when it comes to planting, we just throw the seed on top. And we have people walking over it when they're busy doing another action in the orchard, and it works very, very well. You don't always have to plant, but you've got these, what they call reduced tillage, these planters that just slightly almost slice the soil, as it were. The fifth component, which is always the difficult one, is the animal component, and that's for manure in some form. You have an immediate increase in carbon and nitrogen if you can put an animal component in. It's not always possible, but yeah, it is definitely an advantage. So if we look at those 4, we exclude the animal component. If we look at the 4, in all the cases, you could do it with a cover crop. So you can protect your soil with mulch or compost or a diverse cover crop. Diversity, again, we plant a diverse cover crop on top, then you're going to get a diverse roots at the bottom. The root exudates feed the microbes in the soil, and it is important for carbon sequestration. But back to the point, if you've got a diverse root system, you're going to have diverse exudates, and you're going to have a microbe diversity. The less the soil is tilled, the more stable the habitat for the various microbes. I just want to see, so planting cover crops will cover all 5 or 4 of your soil health principles. That's a mistake there. A diverse cover crop covers the soil, it provides diverse living roots, and it keeps your soil biology alive. When we talk about a diverse cover crop, we usually say include a grass or a small grain like a wheat or something, a brassica, a legume, and a broadleaf, particularly a radish. They work very well in sandy soils and prevent compaction, and then you get that huge amount of organic material afterwards. Depends on which radish you're using. So before you consider cover crops, think about your soil structure. Are you needing to improve your soil structure? Do you need organic material? In other words, are you going to use a cover crop that will give you a lot of biomass, or are you able to bring organic material in from your farm? Do you want to stimulate soil biology or not? Are you wanting to reduce erosion, so something that's going to keep your soil together? Do you need to fix and add nitrogen, and you will then add a legume? You can attract beneficial insects and predators. There are quite a few of the cover crops which have beautiful little flowers. In the fruit, we use Phacelia, very popular. It has a little purple flower, if I remember correctly, but that attracts a lot of the predators. The other thing that you do when you have a cover crop mix is you break your pathogen life cycle. And this is very, very important for you in the vegetable industry. So you're going to say, well, how do I implement this in a vegetable setup? You're obviously going to plant your vegetables, and then you're going to have a row of cover crops, and then you move them on one by one. So if I can say you've got 5 strips, A, B, C, D, E, then A will be your cover crops. Next year you move it on to B, or next season, and so you move on. Remember, your cover crops are also what we call a warm season and a cold season. So you've got to fit it according to the season as well. You can make it out of vegetables as well if you can be very innovative. I just don't know. I'm not that good at vegetable farming, but I've seen this work with some of the big farmers who do vegetables on a large scale, and it can be done. But it's in a small scale. It's not easy, but it's really it's something you've got to try and put into place in your gardens. I'm not going to read through this, the importance of cover crops, but it's a key pillar in your regenerative system, very definitely. The thing that people tend to forget, and I have to add this, is that carbon is released by plants in various ways, and that's why it's equally important to always have a living root system in your soil. In other words, don't farm bare. I get back to that American saying. And then I'm going to end off by how we measure it. There are various measures that we use. The volumetric aggregate stability we do in this laboratory are very basic, especially as an entry level. The microbial respiration test we also do here tells you how much microorganisms or how many microorganisms you have. The only problem with that is it can include your harmful ones as well. But very often we get samples in where there's absolutely nothing. So the soil is what we call dead or very dead. The Haney and the PLFA are very useful, but they're unfortunately done in America at the moment. We're trying to get the tests done here. I've just got to go and find some money somewhere, but I'm hopeful by the end of the year. The Mycorrhizae colonization, it's purely a picture of your root system, and it shows you if you have mycorrhizae or not. And then the nematode community profile is the one that we do quite extensively. This is the mycorrhiza. We will show you these pictures. You can see the opuscule, the little tree, or you've got a vesicle. So it's just a presence or absence. The test is basic like that. It's not easy to do, but it's just a basic answer, a yes or a no. When we look at the nematode community profile, it's quite a complicated test, but you get 3 answers out of it. And the first one is a biodiversity answer. And you can see the trophic groups on the left-hand side. I tell you how many you have of each, and then we put it into a percentage. And immediately, just to read off this one, the plant parasites are too high. There's an imbalance. Your bacteriivores are too many, but it could be a case where you've recently put manure in, a nutrient-rich substrate, and then the bacteriivores can almost take over the whole diversity. There are no fungivores. That's a problem. You definitely need them. The omnivores are present. The nematode predators are none. That's not abnormal. They're quite difficult to find. What's key in this one is your root exudate feeders are very high, and that's usually an indication of stress on your plants, which you can see in the plant parasites. So it makes sense. Okay, and purely by looking at the total number of nematodes, this is 1,460, which is fine. If you have 300 or less, you've got nothing. I've even got 30,000, but that's in a chicken manure sample. But it's only one kind of nematode as well, one of the bacteriivores. So that's also an imbalance. So you can learn quite a lot from the biodiversity. You get this diagram, which is divided into sectors, A, B, C, and D, and you want to sort of go around like that. You've got on your horizontal, on the vertical axis, you've got your enrichment index, and that's an indicator of how much food you have in the system. Along the bottom, you have the soil food web structure index, how many connections there are in the soil food web. So if we look at the soil food web structure index, for annuals, you seldom get it above 50% because you're continually disturbing the soil when you're planting. And then for annuals, we try and reach 100%. On the enrichment index, you're going to be at 50%. And this sounds strange, but I'll explain to you why. So every time you add food like a compost or a manure, it's going to be very high. You've just added enrichment, but it's not composted. So that composting still has to take place, and then it gets down to 50% where you want to be. But then it's going to go down unless you add another manure or a compost or whatever. So it's a very dynamic system. So there isn't actually, you want to be at 50, but remember, you don't want to be below, so then you've got to keep adding. So from that, you can also learn what to do to your soil. For example, we will get, you'll get a point on this diagram. This one, you're in sector D. You have no enrichment or resources and very little soil structure. And then hopefully, if you start implementing the 4 or 5 soil health principles, you will move up and then hopefully up to there. So that's how we use that diagram, that measurement of soil health. That is from one of our trial sites in the apples. We have a lot of mulch, we have a lot of compost as well. Yeah, it's quite a good compost, but you can see on the top, there's also a cover crop which we've put on the side of what we call the berm, that ridge, because we want to cut it and put it between the trees as our mulch. And that's how we use the cover crops. So you can cut it and use that as mulch, and you don't have to bring in from outside. Keep our soils alive. This is in the Sandveld. For those of you who know where our potatoes come from, it's like sea sand. And we started with cover crops, and look at that aggregate which is built up around these roots in the soil. So volunteer rye plants from the previous season, which we used as a cover crop. We wanted rye because it grows fairly high and it protects the citrus trees. So the next year we came in with a mixture of many cover crops, not only the rye. There was a very good reason to start the first year. And then the next year we had volunteers. Okay, and that's called a rhizosheath, if you're interested. And that refers to all that biology that is so active around the roots. And with that, I'm going to end. I'm going to thank Nazeer and Erica and the group again. I've also got to thank the photographers in my laboratory. They're quite good, I must say. And then my contact details. Okay, thank you. I can take some questions. I see Annika has written quite a few here. All right. Yeah. Okay. Erica, I think you must give me the questions. That's going to be easier.
Thank you very much, Sheila. The first thing we're going to do is ask Caroline Kloppert to please ask her questions. Caroline?
Sorry, you want me to ask my questions?
Yes, please.
To just read them? Okay.
Yes, please.
Okay, I've been in a quandary because, you know, I know that you're supposed to cover the soil to recover the biology. And then I heard about fixing algae in the top layer that need light. And they're very important. Have I got the wrong information?
Caroline, I'm not a soil scientist, please, but when we walk into orchards and you have that green layer of algae, and they might not be nitrogen fixing algae, we usually know there's a problem with nitrogen, too much nitrogen. And we say to the guy, you've got to do something here. So I'm not aware of nitrogen fixing algae, but as I say, I'm not a soil scientist, and neither am I knowledgeable about algae either. But it's usually an indicator that there's too much nitrogen in the soil. It's the whole cycling isn't working. I don't know. Sorry.
Okay. I have another problem with the whole regenerative movement in the world and the enrichment of soil as opposed to what Fynbos needs. And how does Fynbos do it? It must have an alternative system that we've got to learn about. But anyway.
Okay. I'm going to have to share my screen again. I'm hoping I can find it quite quickly. I should. I'm going to show you. Sorry, Caroline. Okay. So if you look at this diagram, Caroline, so I had a master's student on about, I can't remember, quite a while ago. It was when we first looked into soil health in the apple industry. I was asked by Hortgrow to lead this whole soil health thing, and it was a number of years ago. I had a master's student, so we decided to go and look at the Fynbos particularly because we wanted to know what would be our baseline in the Western Cape, before we planted the apples. And we did quite a bit of work. And I hope you can see my mouse. So it's in sector B, but at the bottom of sector B. And we were a little bit surprised. You kind of think it's going to be at the other end. But what we realized when we started looking, there's very little leaf drop or organic material under the Fynbos. So again, when we look at these, if we look at this particular diagram, you've got to put it into context. And that's exactly why I've said your aim for annuals is only at 50%. You're not quite going to get to 100 because you're disturbing the system all the time because you're ploughing or disturbing the soil. If you look at the Fynbos, you have no organic material or very little on top. You don't have a lot of leaf drop. But it was in sector B at the top. Sorry, sort of in the bottom left-hand corner of sector B. I hope that answers your question.
What is the name of this 4-part diagram so that I can look it up?
It's called the Nematode Community Profile.
Thank you. That's all my. Thank you.
Okay. Yeah, that was your question, your next question. Okay. Nazeer, your question that Lorata asked about affected by some of the diseases and pests, no, you've got to remove that as fast as possible. And please, when you've got roots that are affected by diseases and pathogens, don't put it into your compost. Go and burn it on a heap somewhere. Because some of those things will go right through, not all of them, but particularly Pythium and Phytophthora and nematodes can go through some of the composts, unless you've got a really good composting system where you're getting your temperatures above 70 or 50 for the nematodes at least. That is a problem. Nazeer's asking a question about the measuring the soil health in a simple way, e.g. the level of the soil organic matter. Yes, Nazeer, you asked me that question in an email. I haven't answered you because the lady responsible for our soil health support center isn't in the room with me. She's quite busy. We have quite a lot of samples that have come in, but I'm going to get her to answer that question for you, because there are various ways of doing it, and then pass that on to Erica as well, and hopefully get it to the group.
Thanks very much, Sheila. There's going to be more time for more questions, but what I'd like to do now is hand over to Dr. Ebrahiema as our respondent to ask his questions. Dr. Ebrahiema is currently a researcher at Stellenbosch University, Department of Horticulture, and co-founder of AquaOrganix. He specializes in post-harvest quality management of fresh fruit and vegetables. He graduated with a degree in BSc Life Science, BSc Microbiology, Masters in Agriculture, and PhD in Food Science. His hobbies and interests include hydroponics, aquaponics, and sustainable forms of agriculture. Dr. Ebrahiema, the floor is yours.
Thank you, Erica, for the introduction. Gladly appreciate it. And thank you, Sheila, for the presentation. It was very insightful and well structured. I'm not actually, I don't know much about arthropods and nematodes, so I have several questions with regards to that, if you don't mind. The arthropods, the Collembola, why are they called springtails? And are they possibly harmful to plants or to humans, or do they carry possibly diseases?
Sorry, I was muted by the host. Okay, now I'm unmuted. Yes, Ebrahiema, so the question, the first one was the Collembola. I'm not aware that Collembola are harmful, and we really need to, if you're going to carry on with the soil biology series, we can get Charlene in. As I say, she's at UCT. They're called springtails because you'll have to go back to the video afterwards. They've got what they call a furca, which is a tail, which is like a spring, and they use it to jump. And that's why when you're walking in the soil, you feel these little things that are jumping up around you, but they're very, very small. And that's, I suppose, movement and whatever reason for. I don't know of any that are harmful to humans or harmful to plants. In fact, in the fruit industry, we've now used, Charlene has got a project going on using springtails as bioindicators of soil health, but they really have a lot of, or they provide a lot of ecosystem services we've discovered. Yeah, and again, the biodiversity in South Africa is quite enormous on the Collembola.
Okay, thank you for the. I have one on nematodes. I read that the nematodes actually causes economic crop loss on your presentation. How as open growers, how can we reduce crop loss with regards to nematodes?
Okay, unfortunately, Ebrahiema, you're going to have to wait for the next series. I'm hoping, because I know when I did the 2 workshops with Nazeer in previous years, that was the one that everybody wanted to hear. And it's probably very, very relevant to vegetable growers. So I'm hoping I'll be able to give that. That you're going to have to twist Erica and Nazeer's arms. And then I give you the different nematodes. I'll give you some of the symptoms and what damage they do, and the control measures that you can use on a small scale. Yeah, I will be providing that information. Sorry, and it's almost for the experience I've had with Nazeer. It's not just a simple. I can't simply answer the question. My apologies.
Don't worry about it. I'm just trying to. Also, how are nematodes actually used as indicators of soil health? Is this based basically on numbers or the
No,
amount of
so
numbers?
it's not based on that. No, it's not based on numbers at all. It's actually quite an intricate system that they use. It was developed by an ex-Zimbabwean, in fact, Professor Howard Ferris is very fond of South Africa. His brother still lives in Durban, but he's in UC Davis. And what we do is we divide them into families and then put them into those various feeding groups, which I showed you. Then in addition to that, we form what we allocate a number, we call it a CP value. On the lower end of the scale is a colonizer. And they come in when there's a lot of nutrients like compost or manure. And they will break that down very quickly. And they're quite a lot of them, they're very prolific. They feed very quickly, they've got very short life cycles. And then as the soil health improves, they disappear and your persisters come in and they've got a CP value of 5. And then behind all of this, we've also got a lot of equations, and then you develop what we call the indices. And I only showed you 2 indices on the graph. You also in the result get a number of other indices, but for the purposes of the talk, I didn't include all those indices that we use. So it's a fairly complicated system.
Thank you. I got just 1 or 2 more questions. You mentioned mycorrhizal fungi. How as home growers, how can we improve mycorrhizal fungi within our soil?
That's also a difficult one for me. I'm not that knowledgeable. There's a researcher at Nitfurbay, the wine guy called Ebrahiema, who has his PhD. And Andre worked in the apple industry, and they're actually, I don't know in your soils, but in the apple industry, they were actually present, but in very low numbers. So I'd imagine if we can get our soil health right, decrease our plowing and tillage and just get the humus right, I think we're going to get our mycorrhizae improved. You can purchase it. There are various, I think there are 2 or 3 products available in South Africa. They come and go. There is a local one developed in Somerset West, and it's sold all over the world, which is quite good. So if you want to, you can. It's a product called BioCult. I know Joe Damas in Rhodes University also has but I haven't seen that product for a while. And they are coming into the market. There are various ones. Yeah.
Thank you. I just got a comment. I remember you mentioned that the mycorrhizae don't form any symbiotic relationship with the genus Brassicus. All right. This could possibly also be due to the fact that Brassicus, they form chemicals also known as isothiocyanates, which basically inhibits or kills fungi. Right? Those who don't know what the genus Brassicus is, they are turnips, radishes, brock-like cabbages,
cauliflower,
yeah,
Brussels, mustards, take quite a few of
yeah.
them. Canola is also
so. Yes. So I was wondering, considering these roots produces these chemicals, maybe I should try this out. Can these roots be used as a fungicide or something to inhibit fungus, like in the extract?
Again, Ebrahiema, you're 2 steps ahead. So that's part of my next talk. Okay. I show small-scale farmers how to do it. We call it biofumigation. And yes. I show them, I've got the recipe. It was done by a student at, a girl from Botswana. She was a student at Tucky's a couple of years ago on how to use cabbage leaves to do biofumigation.
Yeah, because it's quite interesting to me. It's considering we're developing fertilizers and stuff like that with AquaOrganix. So I would probably also look into it myself.
It's a volatile thing, and you've got to have a living system to make it work, unfortunately. but I'll explain that. Or you can, you can phone me if you want to, but if but that's part of the follow-up of the living series is the biofumigation, where we use
That's all from me, Sheila. Thank you. Your presentation was very insightful.
Thank you. Glad you found it useful.
Thank you, Dr. Ebrahiema. At this stage now, Sheila, I hope you've got the energy, but we're going to open the floor for discussion and questions. And I see that there's a couple of questions already that have come through in the chat. One is from Hafiz Sonday, who's asking, what are those 3 organisms that pass through compost heaps?
Okay, so Hafiz, they don't pass through the compost. If you've composted it properly and you've managed to get your temperature above 70 degrees, but there are always air pockets in compost. And the 3 that I mentioned are Pythium, which causes damping off, Phytophthora, which is also a root rot. I don't know how much Phytophthora you get in vegetables. And the 3rd one is the nematodes. The next question is from Nazeer. How does fresh produce imported from neighboring countries impact on local biology? Yes, Nazeer, I'm not going to talk about soil biology because unless you bring rooted material in, which you are not allowed to, the law does not allow you to, we can't even import samples from neighboring countries for testing unless we have an import permit, and it's very difficult to get. The biology, which I'm very worried about, and we have a very good example at the moment, and that is particularly fruit fly. It was called, you'll read about it, Bactricera dorsalis, which is now in the northern parts of the country. There were huge campaigns to keep it out, but insects, yeah, very definitely. And then they come into this country. We have no means of controlling them because they don't come in with their natural enemies. And very often, like this Bactricera dorsalis, it's called the mango fly, fruit fly, I think. Exports from farms which have it, they're not allowed to export to America, for example. It becomes a phytosanitary problem.
Thank you, Sheila. At this point, we do have quite a bit of time available to ask any questions. So please, anybody that is participating, please raise your hand and you're welcome to ask your questions, or you may type it in the chat.
Can you guys hear me?
Yes.
I have one question. Sorry, I'm not sure where to raise my hand on the computer. I'm not used to the computer Zoom. But so there is this, I can call it knowledge around throwing down lots of mulch, where the organisms, they require lots of nitrogen from the soil to break down the heavy mulch that you put on. So I was wondering, if you throw down mulch, do you also throw down like maybe a manure tea for some source of nitrogen for the organisms so they don't have to take it from the growing beds where the crops are?
Hafiz, you're actually asking the wrong person. I should be getting Karina to answer these. If you're putting a mulch on, yes, you do add quite a lot of nitrogen. You can have what we call in the fruit industry, when we really add a lot of mulch, we go through what we call a negative period, and then you can also end up with too much nitrogen. Because as I've shown you with the nutrient cycling, once you get your biology going, then you can really add a lot of nitrogen, and then you've got to go very careful with your fertilizer. Your tea, your compost tea, I don't have a problem with that. However, your often people add external sources like lactobacillus or pinobacillus, and I don't like that. No, keep what's in the soil and what you've got there.
So work with what you have.
Sorry, I missed that question, Hafiz.
Yes, ma'am. Sorry, the connection is a bit breaky, but I got the gist of it. Thanks.
Or write it in the chat. That will help me too.
I did pose another question in the chat. You mentioned 3 things that when going through the compost pile, the making of the compost, these 3 things, they more so often than not don't break down. It's Pythium, and then these 2 other things. It's Pythium, nematodes, and what is the third one?
Phytophthora.
Phytophthora. Okay. I need to make a note of that.
Thanks, Hafiz.
Sarah,
Annika did write that into the chat, so just have a look. It is there.
I see. Thanks.
Sarah, would you like to ask your question?
Sarah. Okay, I can.
Okay, well, my question is there, but so we have a farm where the soil has been very heavily and regularly plowed. The people thought that that was going to be the best thing. It's in an olive grove, but and there's lots of space between the trees. What should we do first to kind of reverse the damage that the plowing has done? I mean, I assume we must plant that cover crop that you were talking about, but maybe there are other activities we could do as well.
Okay, Sarah, I'm going to do a bit of consulting. So how big is your area that you're wanting to cover with a cover crop?
Well, the whole farm is 6 hectares,
Okay.
and the olival is about 2 hectares or 3 hectares. Yeah.
Okay. So because I don't know which area you're in, and I don't know which cover crops are going to suit the area, I would suggest you've got to start with 1 or 2 that you know. So you've either got to ask your neighbors or you've got to ask a seed provider. And the company that does most of the fruit and vineyard cover crops, the mixes in the fruit orchards and in the cover, is Barenberg Seed. And they've got a guy, Ivan, who's very good, and he will suggest 1 or 2. So start off with 1 or 2, depending on what grows in the area and what your problems are, which I showed in that list in the presentation. And speak to Barenberg and they will give you, but you've got to increase it every year till you get to about 4 or 5. You can, in the case of olives, you can plant some pollinating sort of things like phacelias and chamomile and just for the fun of it. The cost eventually becomes the issue. You can't go berserk and just plant things, but that's probably the way I would go if I were you, because you definitely need more than 1. The faster or the sooner you can get up to 4 or 5, the sooner you're going to address the issues of a heavily regularly plowed soil.
Thank you, Sheila. I hope that was an answer for you, Sarah.
That's great, thanks.
Okay, we are being asked by the floor if we can take a 5-minute break. There is time to do that. Sheila, it depends on you entirely.
Yes, you can take a break. You're welcome to do so.
Okay, so at this stage, let's just take a 5 minutes, let everybody go to the kitchen and get some more tea, stand up, stretch, breathe, and please, in the meantime, if you've got any questions, please feel free to put them in the chat. and we'll be coming back in 5 minutes and continuing with the questions and discussions. Thank you. Thank you, Sheila. Please go ahead and answer the questions.
Okay, so I'm going to start at the top. The first one after, after T is from Lloyd. I have a family of mice living in my compost. They seem to work in symbiosis with the other organisms in there. They don't particularly bother me because I would rather have them in the compost than in my garage or house. Is this okay to simply leave it as it is? We have to share with all the creatures in our garden, right? Yes, Lloyd, that's the top end of the predator in the soil food web. So keep them. I just hope they don't become a nuisance. But by all means, use them. As you say, we have to share with all the creatures. The next question is from Caroline. What is it about animal input that builds healthy soil? Isn't planting cover crops and using plant-based compost enough? In my experience, it is not for growing vegetables. Caroline, they've got a huge advantage in terms of increasing your nitrogen and particularly your carbon. And that work has been done on huge farms in the Free State, at the Rates group, Regenerative Ag group. There's a guy called Donnie Slubber, often see him on TV and there's a lot of YouTube, and they've made a huge difference to their soils, the soil health. It is the carbon and the nitrogen. Back to the growing vegetables, it is a problem. We also have the same problem in most of the fruits. They eat the leaves of the trees or the vines. What we do now is we cage them. There are a few farmers who are caging them. It's just a lot of labor. You've got to move the cage up and down in the orchard. And in your case, in the growing vegetables, if you've got space between the vegetables, which you probably don't have, caging the ducks or the geese or whatever it is, the chickens, could work, but it's not always a practical solution. But the fifth component is actually key. The next one, question to please go through the soil food web slide again and just describe the components in a little more detail, please. Okay, now you've got to be patient. I've got to share my screen. And I have to find that slide. I think it was quite high up. Yeah. Sorry, I'm just having a problem with my, there it goes. Sorry, with all the animations, this is going to take a while. Okay. I can't see the question. Sorry, can I just, I'm trying to, okay, so describe the components in a little bit more detail. So this soil food web is divided into the trophic levels. In other words, how the organisms feed, if I could put it that way. So the first trophic level is your photosynthesizers, those are your plants. And then from that you get your waste, your residue, your metabolites from the plants, animals, and microbes, which is the circle at the bottom. And your shoots and your roots. So there's a lot of carbon coming off there, whatever you've put on the soil. And then the second trophic level is the decomposers, the mutualists, the pathogens, the parasites, the root feeders. And those are obviously include a large number of nematodes, fungi, both good and bad, and the same applies to bacteria. It's not as simple as what it looks like in this diagram. Obviously, there are lots of circles which come back, but this is just for simplicity that I've done it like that. And then the third trophic level, again, is shredders or predators or grazers. They feed on other things. So the arthropods will feed back on other arthropods or fungi and bacteria. Your earthworms, which are also shredders and grazers, we know that. You get nematodes that feed on fungi and bacterial, so they're also grazers, and your protozoa that feed on fungi and bacteria. Your fourth level is much higher up, They're very sensitive to disturbance, which is mostly the arthropods that feed on leaves and on other arthropods. They're responsible for keeping your pest numbers lower or reduced, and then your nematode predators, which are also few and far between. But then you've got a very good system if you've already got your higher level predators in place. And then your fifth trophic level is the one above the ground, and that can be anything, the mice, the birds, to a certain degree, sometimes earthworms if they're on top, but they're usually in the higher level predator. I haven't even included them in this group. They are in the third, but they will also be in the fourth trophic level. I hope that's all that you wanted, Caroline. I'm not quite sure exactly what you wanted.
Thank you.
Okay, I must get back to the chats. All right. Chris says, how regularly should a farmer take a soil sample? Excuse me. Hafiz, the question is not quite as simple. It will be, why are you taking the sample? If you're taking it because you've got a problem, you've got a disease or a nematode or a pathogen, whatever the case is, you will take it because you've got a problem, you want to identify it. If you're taking it for soil health reasons, I always say take it prior to an action. So, and then you do it on an annual basis. So prior to adding your compost or prior to adding to your manure, or at the end of its life cycle, you've added the compost, it's been composted, or you've added the manure, or you've whatever you done. So for the farmers who use cover crops, they terminate them at the end, either by cutting it or spraying it or rolling it. That's when they usually take the soil sample. And then you work on an annual basis. Yeah. So in your case, perhaps if you want to take it just before planting to know what you've got, and then you can take it at the end of the growing season, or only at the end of the growing season, then you know what you've got for the next season. The best advice I would give you there too is to go to our websites. We have two, and Annika has included them. It's www.soilhealthlab.co.za and www.nemlab.co.za. The contact details, we have put that, and I've just mentioned them. I can put up the screen again when I'm finished, and you can photograph it. Which nematodes, bacteria, and fungi are important? Hafiz said they all are. But there are thousands, millions of different kinds. It's impossible for us to say what you must have and what you mustn't have. And you can do it with very high-tech DNA analysis, but it's so expensive and it doesn't tell you what it is. So we don't, except for the nematodes, we don't actually count them. We look at how they function. So we also have a system, the PLFA, which you will read, you will see it on our website as well. That gives you some indication as well. But function is more important than numbers in this case. That's why we look at the function and you get that nematode community profile as opposed to how many of each. Erika Nopendula had a question. No, there it is, it's coming up. Caroline, you say you've done soil microscopy and would love to continue learning as a lay advisor to food growers. Can I also learn about nematode classification, or is it licensed? You can definitely learn. It's not easy. At the moment, we only have one person in the laboratory who can do it, but we're busy training. In the past, we used to train in Pretoria. They're not doing training anymore at the Biosystematics Division. So you can by all means train. Yeah, we can try and help you. Well done on the soil microscopy. That I wouldn't even touch it. It's too difficult. Yeah, we've got a plant pathologist who does some of the diseases. That's about it. What is actually wrong with my soil? Because every time we plant spring onions, it becomes rusty. Nopendula, I'm not sure what part becomes rusty. If it's the leaves on top and you've got the rust, I would have to get back to you because I don't have the plant pathologist sitting in the room with me either. And I would have to ask her, and we'd have to make sure what it is. Is it a fungus or is it a bacteria? And that we'd have to analyze in the laboratory. We can look at photographs on a WhatsApp. I don't like to do it. I've been wrong in so many cases that we actually prefer to do the analysis. Caroline, any recommended reading on fynbos soil health? I think we're the only ones who ever did anything, and that was a master's. You can read the PhD. I don't think it's going to help you much. If you're talking about cultivated fynbos, then it's the same principles which apply as what we've done up to now. Thank you for your brilliant talk. Thank you, Caroline. I hope you found it useful. It's actually, this talk is more interesting rather than useful. But yeah, I hope you can do something out of it. Latifa says, my bay leaf tree becomes rusty. How can it be remedied? Latifa, the same as what I said about Nopendula's rust. I'd have to see what the symptoms look like. There's so many that cause rust. It's too difficult just to tell you. Annika has posted the details for the Soil Health Support Center and not Nemlab. The telephone number is the same, and the Nemlab is simply www.nemlab.co.za. Latifa, you're back to the leaves. Yeah, I think I answered that. Namondi said, how much will the soil test cost and how much should one send? All those details, how to take samples, what the costs are, and which tests you're wanting done, look at both websites. So the Soil Health Lab only looks at soil health. The Nemlab looks at the damaging fungi, bacteria, nematodes that we test for. And each test has a different cost. I'm not too sure if the plant disease costs are up yet. I doubt it. We haven't quite got to that point yet. We've only just employed the lady down here in the Cape. We have a lady in Durban who does it for us. Anika says, I can send the price list to someone who can send it out to all. We need at least 500 grams of soil for an analysis. The price list and sampling guide is also on our website. Thank you, Anika. Okay, Nomonde says, thank you. Sarah says, can you say something about types of fungi which are not welcome and how does one recognize them? If one can without a soil test. No, Sarah, you can't. The only way you can recognize them is looking at the symptoms, be it on the roots, be it on the soil, but it's not that accurate. Some of them are very good and others aren't. I'm afraid that's just the bottom line. They're all microscopically small. Some of the symptoms overlap and some are similar. Yeah, I mean, we have a lot going through here and it's certainly not something you can just do. The best would be, and there are a couple of books on diseases. Years ago, the University of Natal had a book. It's very outdated. I have it in the laboratory. I don't use it. And you're more than welcome to have a look at it. I can, we can work on it and produce another book. It's very basic. Yeah, on how to control, come to think of it. So if that seems to be quite an issue and, somebody's got time, they can draw up a book like that. The rust on plants might be due to water. If watering with well-point water, it is high in iron and other undesirable elements. So that could also be contributing to the rust. I get back to my point. We'd have to look and see what is causing the rust, in the laboratory. And if we don't pick anything out in our test, which we have to put on a Petri dish, well then it could be the iron. Abida asks, my question is about the soil sample we will send. I will chat to Erica about it privately. Abida, feel free to contact us and ask us. Yeah. Okay. Nopendula asks about the rust again. Rust is a difficult one. There are lots of pathogens that cause rust. Corin says, I disagree. Your talk is very useful. It gives us a great place to start further investigation. Yes, Corin, that was my point. It's just a place to start. It's the basics, and that's what Erica wanted. But I'm glad you found it useful. Hafiz says, should your soil be fungal or bacterial dominated or a combination? Hafiz, I don't want to get into this debate. It's an Elaine Ingham debate, and I don't really agree with it. Yeah, it's a difficult one. We do in our test tell you whether your decomposition is fungal or bacterial. If I get back to the colonizers and the persisters, which I was explaining to somebody earlier, I think, yeah, I was explaining to Ebrahiema about it. If you're going to add raw manure, it's going to be bacterial dominated, and there's nothing wrong with that. And eventually, as the succession takes place on your structure index, it's going to become fungal dominated. But yeah, that whole thing, I'm not too fond of that idea. Caroline says, sorry to ask again, what was the name of the student whose thesis is on fynbos soil health? Yes, what I've done is that I've asked Annika sitting next to me, she's, I think, sending you, oh, there's lots of messages. I think she sent you in the link in the chat, she sent you the thesis. I might have a copy in my office. I probably do, because I was a supervisor, but it's a fairly thick document and very, obviously very technical, but you're more than welcome. And I seem to record that we would have written a popular article in an essay fruit journal, but that would have been a number of years ago. And then Caroline also says, thank you. You agree with Corin and looking forward to the rest. Yes, the next one is very practical, I must say. Corin says, could you recommend a reasonable microscope to use? What is the best magnification level for nematodes? Corin, we get this question quite a lot from people who are following Elaine Ingham's tests. My best advice to you is come to the laboratory. It's difficult. I can't, tell you, yeah. There are lots of different microscopes. It depends on what you want to do. Rather visit the laboratory, come and look what we have. We have lots. We use them for lots of different things. I think you must rather come and have a look. Yeah, that's the best advice. Fawaz says, Sheila, I really love the information that you are sharing. Again, thank you. Thank you. Glad you're all finding it useful. And then Annika has added the fynbos thesis for you, Caroline, and she's listed the name of the title of the thesis. And as I say, those things are usually available. I see it was 2013 already, but as I say, yeah, you're more than welcome. Ray says, can one visit the lab and could I bring my 8-year-old? Ray, you're more than welcome. We've had school groups here before. Biology groups or whatever, that's not a problem. Not too big a group though. Ray, you're more than welcome with your 8-year-old son. It's not as exciting as one would like to think, but it is, yeah, you'll see quite a bit. Probably best to phone ahead. We might not be here, or we're all busy with something, or we've got a huge meeting. But yes, you're more than welcome to come and visit.
Thank you very much, Sheila. At this point, Dr. Ebrahiema, do you have any further comments?
No, thank you, Erica.
Okay, thank you very much for your participation. And thank you
too.
very much, Sheila, for answering all of these questions. In closing, Nazeer, would you like to ask your questions?
Erica, Nazeer has just posted a question.
Okay, go ahead, Sheila.
Yes, Nazeer, you can assume that you're improving your soil health if you're following the 5 soil health principles. But what I found just with the farmers around the Western Cape, oh, excuse me, what they're finding very difficult is you're doing something, but it's in the dark. And that's why the measurement became an issue. And that's why that part of the laboratory is growing so fast, is people want to know, am I on the right path? Am I actually doing something? Because you really are, it is in the dark, it is underground. You kind of wonder what you're doing. So the principles are very important. It definitely improves soil health. I can add to that, Nazeer, we've got trials running in the fruit industry, which we did for 4 years on two sites, one in the Collembola, one in the Warrenbokkerveld, just to get different climates. The one was on apples and the other one was on pears. The one was ridged, but we had control plots as well. It's very difficult to have a control plot when you're planting cover crops and you've got seed floating all over, but the measurements definitely showed an improvement. We looked at a lot of things. We looked at half of which I haven't even mentioned, but we looked at cover crops, how well they grow, how much biomass. We measured all of that. Yeah. So we can, even for that matter, if I can twist Matthew's arm, he's at, he works partially for Hortgrow and partially for the university. He's at conservation ecology and entomology, but he's an entomologist, and he has some good results from his trials, but that's commercial scale on apples. I'm quite sure Hortgrow would share the information.
Thank you very much, Sheila. I think at this point we are now at nearly 2 hours. We're going to have to close now. Like you have said, this is just the beginning of the Living Soil series. Today we've just covered the basics, and we anticipate to be having at least 2 more sessions. We are going to be sending in soil samples from 5 different growers in 5 different places, and our next session will go into the details of your analysis on the nematodes, on that, and how you deal with the diseases. So thank you very, very much for your time today. Nazeer, is there anything you'd like to add?
No, just to say thank you, Sheila, and thank you, Erica, for putting this together. I know behind the scenes, a lot of hard work that goes in organizing something like this. And I'm looking forward to the next session already.
Great, thank you, Nazeer, and thank you to everybody that participated today. You will be getting an email with all the details, the presentation, the link to the video, and any other details that you have asked for. And we look forward to seeing you at the next one. Thank you very much, everybody.
Thank you.
Thank you, Erica and Joe, for setting up the workshop.
Thank you, Dr. Ebrahiema.
Thanks so much, guys. Enjoy the rest of your day.
Thank you, Hafiz. Bye-bye.
Cheers. Bye.
Bye.